Okay, so good evening, everybody, and I welcome you all for Council for Research Ayurveda General Club meeting for today. And our today's presenter is Ashwini Godbole, ma'am. I would like to introduce her briefly because her CV is very extensive. But I just wanted to tell you that she's a person who is already working on a multidisciplinary level. So she is currently working as assistant professor at Center for Ayurveda Biology and Holistic Nutrition (TDU). Almost since 5 years or 6 years odd, she has been working as a postdoc fellow at IIM, then principal investigator at FRLHT trials, and today at assistant professor at TDU. Her trainings are very extensive in terms of study of mode of action of Rasayana with major focus on aging and neurodegeneration. Also, her postdoc project was focusing on anti-helminthic activity of plant drugs such as Vidanga. And so it is like an anti-helminthic as well as a Rasayan herb. So she wanted to explore that aspect. And various trials she has undertaken for her PhD project as well as her MSc project, majorly relating to various sort of herbs and botanical aspect. There are publication in number of publications with her, but the major publication I would like to focus on expressions and purification, and also in terms of evaluation of traditional herbs as substitutes for various models in the animal trials scenario. She is also having many achievements in her hand, like almost 10 plus publication in international peer reviewed scientific journals, many oral presentations at international conferences, and DST SARB grant. She has been awarded with a research grant for young scientists. She has also been awarded with the best poster and many times also with Sarojini Damodaran International Fellowship for undertaking a short-term project in the laboratory of Professor Russell Jones in California. Her CV is really so extensive that I just wanted to focus few key inputs from her CV, that even if she belongs to overall biotech or botany or basic sciences, her major interest is towards evaluating the herbs and how they can be revalidated in terms of the medicinal aspect. So today her presentation is also about that. And without wasting much time, I would like to call her and deliver her presentation. And this journal club meeting is also unique in terms of evaluating a journal article, so that next time we try to write the article, we know how to put forward our research so that everybody on the global platform understands, and that becomes a very key step to also plan our future researches. The Journal Club activity for Council for Ayurveda Research USA, and in India as a India chapter, we focus on taking all sorts of multidisciplinary areas in hand, so that many students are also having that collaborative approach, and they will also take future projects on the similar ground. So thank you so much, Madam, for accepting our invitation today, and being here in your busy schedule. Is, I know it's a Saturday evening and you're taking out time for us. Thank you so much for that. You can share your screen, ma'am.
Yeah.
Yeah.
Thanks for the introduction. And I'm actually very young in the Ayurveda biology field, even though I've been working in other fields. Yeah. So,
yes, visible.
Yeah, visible, right? So,
Yeah.
it's a great pleasure to present this paper to in CAR Journal Club. One, because this paper shows that how we can meticulously actually look at a drug or a plant extract which is from and which has actually roots in Ayurveda. It is taken completely from Ayurveda, not just by chance but by design. Ayurveda uses this in the conditions like Alzheimer's or age-related problems, and how we can actually look at it systematically, what it does, what is inside the body, and how it can do. So, Why should we be bothered to study Alzheimer's disease? Because as you know, you must be knowing, you can read and you can see it, that one is that the lifespan has extended. Global lifespan has extended quite a lot. It's much higher, and thanks to all the vaccines and antibiotics and so on and so forth, infectious diseases. Though we are talking about we are in Corona time, COVID time, but in general have become much life threatening, much less life threatening. And but at the same time, lifestyle diseases and life age related diseases are on increase, and that's why we need to look at this one age related disease, which is Alzheimer's disease or dementia, is actually growing every day as we are speaking. So that's the problem, and you can see that almost 50 million people have that problem. And in 2001 to 2040, there will be 100% increase in the global cases, and 300% increase in countries like India and China, or South Asian countries, which are developing countries. And this is the seriousness of this disease, and it can actually bring down the quality of life very drastically. And that's why we need to look at it. And it's not the medication per se, but the cost of care is very high. And you can see the Alzheimer's is actually connected to dementia, that is short term, mainly short term memory loss, and then eventually a lot of memory loss, like even the long term memory loss, and then recognition, attention, everything goes down. Disorientation, mood swings, problem with language, loss of motivation and self care, and inability, and serious behavioral issues. And as you can see in the later portions, you will see that this will obviously lead to increase in cost of caring. And cost of caring is something which is not normally considered as a big cost of medication, cost of surgery, cost of post surgery care, and all such care costs are considered. But this is a very big cost, because the person really cannot take care of himself or herself, and we need to really uh, have a complete 24 bar 7 caretaker for the person, either from the family or otherwise professional. So this is why it becomes very important to study it. And what happens during Alzheimer's, you can see on the left hand side, you can see of the screen, you can see there is healthy brain, and the brain definitely shrinks. The shrinking is happens anyway during aging, but this is very prominent in Alzheimer's disease. And why does that happen? The shrinkage happens because there is death of the tissue. That's why the death of the cells, and that's why the shrinkage happens, and the gaps, the ventricles between the brain start enlarging because there is death of the tissue. And you can see that the brain nerve cells, which are nice and connecting each other and passing the signals through the neurons, one neuron to other neuron, now start getting disease. That means there is aggregation of protein like amyloid. Here there is garbage protein, and then there is inside the cell, and that there are proteins outside the cell getting accumulated, and inside the cell as well. And that actually makes the cell very very sick, and eventually the cell dies. And that's why the neurons start dying, and that's what leads to the shrinkage of the brain tissue, and as well as loss of memory, because the synapses break away and the shrinkage, memory is lost. Why does that happen? The A beta protein, which I talked to you about. So there are mainly two proteins involved in this, which people have seen over decades, that there are two main proteins. One is the amyloid beta protein, which aggregates aggregates outside the cells normally, but there are oligomers inside the cells. And the other is tau protein, which is actually both these are our own proteins, and they are always there in every human being and everybody at all the ages. But suddenly something happens, and the problem starts. The A beta protein, amyloid precursor protein, as it is called as APP, actually you can see that if gets cut by alpha secretase, it is in the soluble form, that is soluble APP. And if it gets cut by beta secretase, that base, then it forms actually one soluble portion, that is in green color, and other portion which is in the red color, which start accumulating, and that is called as A beta protein, and it is made because of beta secretase, so it is a amyloid A beta protein, and that starts getting accumulating because that has a tendency, those fragment of protein has tendency to aggregate, stick to each other and make big blobs, or so that's the problem. That's what starts actually, that's what the beginning of the problem is. Then this A beta, which are aggregates, actually start damaging the cell wall, cell membrane, and then also the organelles inside. And that creates very high amount of stress in the cells, physiological stress in the cells, and the cells die. So what do we see in this paper then? So how can we see what happens actually in the brain or human beings? So there are models which we can see. We can take part of Alzheimer's disease, what we know by research, that there is tau protein entangles, and then there is Alzheimer's protein, A beta protein, which is getting accumulated, and then you express it in the model organisms and try and study that, because we cannot see anything naturally spontaneously happening in large number of organisms. So what we do is that take human protein, put it into the uh, uh, put it in mouse or any other model organisms. In this case, for this paper, they have done it in mouse. And what is done is that there are three types of models, but what we are going to talk about is the middle one, and that's the one which the paper authors have used for doing all the experiments. And in that, there are presenilin is one other protein which collaborates with APP, that is amyloid beta protein precursor, and that together actually causes Alzheimer. There are many other proteins also, but these two are one important ones, and that if it is expressed in a mouse, then it will cause Alzheimer's as the mouse ages. So it mimics many aspects of human Alzheimer's disease. So that's what the model is all about. And as you can see, this is a mouse brain, and you can see this portion here, inverted U shapes portion here is hippocampus, and that's involved in the memory of working memory of for the mouse also. And then there are three regions: frontal cortex, uh, hippocampus, and the cerebellum is where the gene is expressing. But the main problem happens in these two regions, and cerebellum actually does not, expression in cerebellum actually does not cause too much of a problem. That also gives us a hint that the same protein in some region of the brain causes more of a problem than in other regions of the brain or other regions of the body, because the whole body has this protein, whole organism has this protein, but it actually causes problem mainly in brain, and that too certain areas of brain. So these are some important facts we should keep in mind while we design the experiments, or while we design the model by itself, or while we select a model, that what exactly happening, which part of the disease is actually getting replicated, how far is it getting replicated, what are the limitations and what are the advantages. So that's how we should select the model. And this is one of the best available model to study Alzheimer's disease currently. So you can see how does this mouse behave. It behaves like as it ages, it keeps start showing Alzheimer's kind of behavioral deficits. As you know, as the person progresses in Alzheimer's disease, they will have disorientation, they will not be able to find their way, spatial memory loss, and so on and so forth. And that is mimicked behaviorally in this mouse experiment, where this experiment is 8-arm maze, and where there will be bait at some places, and some other arms will be closed, and the mouse will be trained in this maze, and then there will be test run, and there will be test recording. So that, and you can see one group of mice was treated with vehicle, which is the normal solution, saline solution. The other group of mice got the extract. These are both transgenic mice. So you can see TG vehicle and TG extract here. And so extract is ashwagandha extract, that is ethanolic extract, which is actually has a lot of concentration. It is concentrated in withanoids. And these components of ashwagandha extract is actually concentrated in this ethanolic extract of the of ashwagandha, alcoholic extract of ashwagandha. So you can see that the normal you can see that the wild type mice mean normal mice, there is no Alzheimer's to this, so it's normal behaving normal mouse. You can see that there are errors which happen, and in the errors there is decline in the errors as you treat the so the errors remain constant. The mouse keeps doing errors, and in fact it starts increasing as the vehicle in the transgenic mouse which has Alzheimer's. Whereas the in the Withania somnifera tree extract treated mouse, the errors are much less. So you can see for all the types of errors, the errors are much less in the middle-aged mouse as well as the old-age mouse. Because in this mouse model of Alzheimer's disease, as the mouse ages, the severity of the disease increases, as it would happen in human beings as well. So you can see that in both the cases, the transgenic mouse, which is treated with extract, which is open circles here round, you can see that the behaviorally they do much better than the other the normal either untreated transgenic mouse. This mouse which is treated with extract does much better. That means it can, the behavioral advantage is definitely there. Now once we see advantage at the behavioral level, then you can go deeper with this kind of experiments. You can go deeper and deeper. And why, one thing why I thought I would select this paper is because if you see that many of the MD thesis also, because there is a limitation on time, many MD students choose to work with animal models. And you can see in this paper what depth these authors have gone into to study this one particular event. That is that if Alzheimer's is caused by transgenic gene expression in mouse, how does Withania somnifera extract whether it can stop it? If it stops the decline because of Alzheimer's disease, then how does it do it? So these are the two main questions, and they are trying to understand all this with taking lot of different angles and doing lot of experiments. And so first experiment, as I said, is a behavioral experiment, because finally the behavior is the major thing. You can see how that at the behavioral level the extract is protecting, the deficits are not setting up out. So that's how. And you can see the extract treated mouse does as good as the normal mouse, which is not Alzheimer mouse, normal healthy mouse. So that's how they are, that's how effective the extract is. So that's what this, in the middle age, the mice which are normal mice and the mice which are treated with extract and being Alzheimer's mouse, both behave very similarly at 30, 28 hours and 28 weeks and above, 28 days and above. What can we see? Once we see this at behavioral level, then if we look at it at tissue level, you will see, can you everybody would appreciate that this is a vehicle, means the control, the Alzheimer's mouse, which is not treated with extract, but it is treated with the media alone, just the vehicle. And you can see that there are these dot like things here which are present, and the extract, in the extract fed mouse brain, you will see these dots are completely missing. These dots are A beta aggregates, and there you can call it as garbage, or you can call it as the toxic protein aggregates. And you can see these toxic protein aggregates are much higher in the vehicle group, in the middle age as well as in the old age mouse. And in the hippocampus, cortex and hippocampus region, you can see a lot of aggregation of protein, and that which is toxic to the brain. And you can see in the extract, it's almost not there in the middle-aged mouse, and it is quite a low amount in the old-age mouse, in both the regions, in the hippocampus and in the cortex region of the brain of the transgenic mouse, transgenic Alzheimer's mouse. And you can see the quantification of the same data here. If you can calculate percentage plaque area, plaque is the protein, toxic protein aggregates, and you can see that in the uh, transgenic mouse which is treated with extract, it is almost nil in the middle-aged mouse, and you can see mice, and then you can see in the old-age mice that it is actually drastically reduced in the brain, hippocampus, and the cortex region. You can see that the treatment with you can see the quantification, and then you can see the tissue. Now in this, this is a staining experiment where the antibody has marked the A beta protein aggregates, same thing which you can see by getting the extract of the brain extract and running, and you can see all the forms of this A beta protein, that means monomer. As you, if you remember that my earlier slide here, you can see that there are monomers which are not toxic, means single unit of the protein. As it starts aggregating, it forms oligomers, dimers, trimers, tetramers, and then it starts aggregating in larger and larger numbers, and that's when the problem starts. So you can see that the A beta protein, you can see that the vehicle and the extract here in the middle-aged mouse, and the wild type which does not have any transgene, no protein, no Alzheimer's protein, it's blank here. And in the vehicle control mice, you can see that there are monomers and higher amount, and then there are trimers and dimers and trimers, tetramers, and that's what we can clearly see it again and again. In the staining method also we can see in the, this is called as Western blot, and Western blot is where you can separate out by size. Here we are not able to see the size difference, right? Here we can see with the monomers and dimers and trimers and tetramers separately. So you can also see how the protein is getting aggregated. So you can see the trimers and tetramers are higher amount in untreated, no extract mice, and then brain, and then with even extract, when it is treated with extract, there is some monomer but which does not get aggregated. At the old age also, the aggregates are very in high amount in the vehicle treated mice and brain tissue, and but it is in lower amount, in the much lower amount in the extract, Withania somnifera, ashwagandha extract treated mouse. So now more, so you can see monomer, dimer, trimer, tetramer, everything is reduced. Then one can say that actual the basic protein, that is APP itself, is not there. That's why in Alzheimer's mouse, that's why maybe this is happening, but that's not true. You can see the APP levels are almost same, and this is how the experiments with the model systems are done. You always have to have something, some control. You will have to have something which will say that this whatever event which you are saying is because of what you are, the mechanism which you are thinking or assigning it to, and the basic mechanism exists. Like the transgenic mouse will always produce APP, the basic protein, amyloid precursor protein, but the further aggregation and further problems do not happen when the mouse is provided with extract or fed with extract, Withania somnifera extract. And you can see that this is a time dependent, process. That means in the vehicle, of course, there is lot of protein aggregation. As you go ahead, 7 days of treatment, it reduces. You can see it in the Western blots again below. You can see 7 days it reduces a little bit, then 14 days there is further reduction in aggregation, then at 21 days you will see really less aggregation, and then you can again see the graph here, the quantification of that. And again, the beauty of these experiments is that you can actually quantify this data. You can quantify intensity of the plaques, the area here, the brown one, and you can quantify the intensity of the band, the black intensity of the band, and then you can plot it. and that's relative optical density and versus the number of days the treatment has been done. And you can see the transgenic mouse which is treated with vehicle normal solvent versus the extract, and you can see the monomers have reduced, the trimers have reduced, the trimers and tetramers, everything is much less in the Alzheimer ashwagandha treated mouse, and the APP but APP remains constant. So this has actually shows that how over time it is doing it. So the ashwagandha is reducing the plaque load, and that's why the behavioral is getting better. All the pathology is actually getting reduced. A beta pathology, and very beautiful result this is. One should pay attention to this. We can see again and again the authors have made sure that this is not artifact. Whatever has to happen, whatever we should see, and all the points are matching, and we can see the results in that way. You can see that A beta protein in the transgenic mouse, which is treated with just solvent, remains constant, but in the extract treated mouse, it is reducing. All this in the group where it is again, it is constant in the older animals also, and it is reducing in animals treated with extract. At the same time, where is this protein going then? If it is reducing from the brain, it is going somewhere, right? So it is going actually in the plasma, and you can see that there is increase in plasma A beta as when in the treated animals. Extract treated animals, you can see the open circles, and there is increase on the day 14 where you start seeing the decrease, there is increase in the plasma. That indicates that the protein is coming out, the bad protein, the toxic protein is not getting accumulated in the brain, it is thrown out in the plasma and it is getting cleared. So this gives us a clue, this gives the author a clue that what must be happening, what must be the mechanism of action of ashwagandha extract. And then they further started digging it up deeper and deeper, and then you can see that there is uh, uh, lipoprotein like receptor protein, LRP, and then there are other proteins NEP and soluble LRP, and these are the proteins which actually help uh, exit of the toxic protein from the brain to the plasma. So this is how people have checked how this can take place, and here they have said that in whenever there is animals are treated with Withania somnifera, you can see that the LRP in the vehicle it is much less, but as you go on 7 days, 14 days, 21 days, 30 days, you can see this lipoprotein like receptor like protein which is responsible for taking out the bad toxic protein out from the brain and into the plasma goes on increasing in amount. So the expression of this protein goes up over days. Same in the middle age as well as in the old age animals when treated with extract. And you can see the NEP, neprilysin protein, which is also involved in this kind of trafficking of protein outside and precursor of NEP, LRP. You can see that that amount is also going up here. So this is how the authors have systematically dissected out the pathway and how it must be working. LRP is a liver protein, and LRP also is present everywhere, but this LRP is actually in the liver, and then that is NAP is also in the liver, and soluble LRP is deposited in the plasma. And where does soluble LRP then end up? It ends up at the blood-brain barrier, and this is very beautiful result. You can see that the soluble LRP which comes from the liver to the brain, and it is actually at the epithelial cells, and the epithelial cell marker they have used, you can see green color here is indicating LRP, the lipoprotein receptor like protein is stained in green here, fluorescent green, and you can see it is much more in ashwagandha treated. You can see the fluorescence is much more in ashwagandha treated animals, and it is counter checked where this protein is, where is this fluorescence, it is in the epithelial cells and it is in CD31. CD31 stain, counter stain shows that this is the place where it is. It's in epithelial cells here, and you can see it is, when you merge it, you will see that it is correctly at the same place. And the same LRP protein is at the borderline of, or you can call it as, we call it as blood-brain barrier, right? So it's at the blood-brain barrier. It is at that correct position where it can take the toxin protein from the brain and push it into the blood. But that same LRP protein is not present in neurons. So you are confirming that whatever results one author has confirmed, and whatever results they see at the epithelial cells is the real result, and you do not see that kind of change in the neurons. And once you stay in the neurons, you won't see, but the blood vessels which are actually present in the brain, all the regions of the brain, especially the in the cortex, you can see that the blood vessels are lined by the blood-brain barrier. These structures, which are elaborate structures, which are called structural entity, which is blood-brain barrier, and these are lined by uh, LRP, the lipoprotein receptor-like protein, which actually is involved in pushing the bad protein out from the brain. Yeah, so again, you can say that this might be just by chance. So what we do is that then you can confirm that what is happening in the brain, the LRP is not going up, it is remains constant. In the liver, once when the animals have fed with ashwagandha extract, the liver starts producing more of LRP, and you can see very clearly here LRP, the RNA, the one step before protein. You can see it is same in the treated and untreated animals. In the brain, it is same vehicle and extract treated animals, while in the liver, the extract treated animals show a large amount of LRP. And same in the older animal, NAP, the related protein, also there is higher amount in the liver as compared to in extract treated group as compared to the vehicle treated group. And you can see, if you quantify also, you will see the similar results. At the when the LRP is going up, simultaneously the A beta is going down. So the A beta, which is the toxic protein, you will see that A beta actually goes down in the middle age animals as well as in the old age animals. And you can see there is a negative correlation between the brain A beta 42, and NEP in the liver and LRP in the liver, and soluble LRP in the plasma. So you can see these yellow marked cells in the first column, where it is brain versus the LRP, NEP or soluble LRP. You will see there is negative correlation and the values are negative. While for NEP to LRP, they are positively correlated, and LRP and soluble LRP is also positively correlated. So it clearly indicates that as the LRP, so lipoprotein receptor like protein is the main thing which is getting induced and getting overexpressed in the liver, they going to the brain and sitting at the blood-brain barrier and clearing out the plaques. So now the final confirmatory experiment is this, and it says that now we want to confirm once again that when the liver LRP is the factor which is actually leading to protection because of ashwagandha extract, protection from Alzheimer's. Then you can see there is a technique which we can use in the molecular biological technique, which is called as antisense RNA technique. So if you give antisense RNA and to the express antisense RNA in the animals which are already expressed, are already fed transgenic animals, that they are Alzheimer's animals, and now you are giving antisense of liver LRP which is in liver form of LRP. That means the LRP will get shut down, the LRP won't get expressed, and you can see that here in the brain the LRP in the normal gene and the antisense gene, it is remaining constant, but in liver now LRP amount has gone down, and here also you can see the LRP amount has gone down in antisense group, the group which is also expressing antisense gene, of LRP. You can see everywhere that the soluble LRP is also going down, because if LRP is not there, the soluble form also won't be formed, and you can see the antisense LRP has gone down. And then you can see that in that case, Withania somnifera is not effective. So A beta protein, here you can see A beta protein, which was nicely getting cleared earlier with, you can see here again, it was nicely getting cleared because of the Withania somnifera extract. There was never a problem. It was getting into plasma and getting cleared out from the body. Now stops getting cleared because there is no LRP, liver LRP here. You can see A beta in the normal transgenic thing and antisense expressing transgenic mice, it is same. That means now A beta is not getting cleared off. In the plasma also, it should go up. The A beta should go up. So that's what we had seen here, that in the plasma the A beta goes up, A beta level go up. But in this case, that does not happen. A beta level in plasma remain low, and that means the Withania somnifera, which earlier was helping and it was to reduce the A beta level from the brain, and actually behaviorally also it was taking care of the behavioral deficits because of A beta, has now stopped working because of the antisense, because the LRP is not getting formed. Excuse me. So the model is that, one second, I think I missed one slide. Yeah. And then just to add to the story, one more layer to the story, authors have shown that there is another type of cell in the brain that is called as glia. So you can see in this picture, you can see there are astrocytes and there are glia. These are immune cells of the brain. So there are neurons. We all know neurons. They are the nerve cells, and they are the cells which are which take care of main function of the brain and the nervous system. But then there are other types of cells, and these are kind of immune cells of the brain. And you can see astrocytes, which sit right at the blood-brain barrier, are also important to control entry of what is coming inside the brain and what is going out as well, but mainly what is coming inside the brain. And microglia are another type of immune cells of the brain, and they are actually taking care of, again, maintaining the environment of the brain clean and nice, so that there is no toxin getting accumulated. So you can see a very beautiful result here, that the green fluorescent protein here is actually a beta protein, and the red ones are the microglia or the soldier cleaner cells, and you can see that the red cells are now trying to get around the toxic protein, the garbage protein, and trying to clean it off. And as you give in the vehicle, they are not able to do it. There is a lot of protein getting accumulated. There is a lot of garbage protein, toxic protein is getting accumulated. But as you feed the animal with ashwagandha, you will see slowly these green garbage protein is going away, and the cells are actually the cleaner cells are actually taking care of these green garbage protein and going away. And then as the toxic protein is cleared, the amount of cells in that area, the number of cells in that area, the soldier cleaner cells in that area also reduces eventually, because you don't want the microglial response to be continuously there. Once the work is done, they also reduce. So this is very nicely shown in this paper. So finally, if you have to summarize the paper, then we can see that there is in the brain, there is accumulation of A beta protein, which is depicted in yellow here, and something happens that the protein which is also sitting at blood-brain barrier, also goes up in Alzheimer's condition, and it actually starts putting back the toxic protein back into the brain. But when we feed ashwagandha to these animals, or then the liver starts working, it produces LRP, and soluble LRP form also and the LRP1 goes and sits in the blood-brain barrier now and pushes out all the A beta out from the brain and into the plasma or into the blood flow, and then soluble LRP bound with A beta is again taken care of by the system and it is excreted out. So that's how a whole mechanism of action of Withania somnifera extract can reduce the A beta or the Alzheimer and related problems in the transgenic mice. And similar, so when we say it is happening in mice, we are mimicking certain portions of Alzheimer's disease which are related to human Alzheimer's disease or human pathology. And if this is happening here, then it's very likely that the similar thing would happen in human beings, because these are also mammals. The kind of pathological effects which we were seeing because of A beta, loss of memory and all those things are also replicated in mice model, and that's how we can actually start deciphering how it must be, how ashwagandha must be working in human beings, in human body. So there are certain limitations. Of course, the model is not completely mimicking what happens in the Alzheimer's disease patients. It is restricted only to A beta. There is no tau protein, and then the other factors are not taken care of. A second and major thing for us, people who are connected to Ayurveda, is that ashwagandha formulation. Maybe ashwagandha aristam is also made, so we can call it as ethanolic, the alcoholic extract, but still it's not mimicking the formulation as such. How ashwagandha as whole formulation, how it must be working. Part of it is used. So it's only the giving us information mainly because of part. Who knows, maybe if the whole formulation is used or whole traditional formulation is used, there might be added advantages as well. And and another thing which I feel is lacking in this paper is that there is no data about how the aggregation can be reduced. So one is that the A beta aggregates which are there already in the brain are getting pushed out. The A beta protein which is made in the brain is getting pushed out. But how is there anything which ashwagandha is doing to stop aggregation of the aggregation of A beta is something which is not addressed in this paper. So these are certain limitations of the paper which I feel, and the rest all, I think the model is quite good. It has given a lot of insights, and author has acknowledged the Alzheimer, the ashwagandha, which is knowledge from Ayurveda, and we can actually look at the say like Ayurvedic advice for such things like dementia and make the design the treatments accordingly. So authors have acknowledged that, but apart from that acknowledgement in the introduction, later as such the formulation is very restricted to the alcoholic extract, and that to semi purified alcoholic extract, and the whole formulation is not uh, is not discussed further in at any point in the paper. And the other aspects which actually when this paper got published, it was it's kind of it's been many years now since I think in 2011 is when this paper was published, but after that the field has gone ahead and field has progressed, and now especially now it is becoming more and more clear that this blood brain barrier which we talked about, where the LRP is functional and liver makes it, it goes to the blood brain barrier and works. So everything whatever is neurotonic or nootropic or taking care of neurodegeneration or as a medicine for brain function need not work only at the brain level. There can be many places where it can work, and one of the places which we saw in this paper is liver. And similarly, there are many more and more data which is coming up, which shows that the another site which we need to really be taking seriously is gut, and that's what Ayurveda says, that we have to take everything is related to gut and how gut is functioning. So we actually can see that there are lots of probiotics, the good gut microbiota, which helps in health, keeping the brain and nervous system healthy. There is increased factors like neurotropic factors, good for nervous system factors like BDNF, which get made, and actually the brain remains healthy. And then if there are antibiotics, or even if there are bad bacteria, then that also actually causes a problem. So there is neuroinflammation, there is reduced BDNF and reduced NMDA receptor function, and that's what actually causes problems in cognition. That's why our brain is very closely related to gut, and we should take care of it. There are added data which we can read in the literature, is that if the gut is leaky, the blood-brain barrier is also leaky, likely to be leaky. So risk of blood-brain barrier being leaky. So if you can see, if this blood-brain barrier is leaky, then lot more toxic compounds can come enter the brain region, and that can be really a problem for brain function, and that can cause inflammation, that can cause toxicity and death in the brain, and also on. So what we can see is that tighter is the gut, if the gut is leaky, the blood brain barrier also tends to be, the risk for blood brain barrier to be leaky also goes up drastically. So it is required to maintain the gut health, and that's what Ayurveda says. Vata is the main dosha for this, and gut is the main site for Vata. So the large gut. And you can see that that's how we can actually bring in these concepts together, and we can design our experiments and complete them and get insights from those experiments, which might not give us the complete picture at once, but it definitely fills in the important gap in the understanding. And as we start filling these pieces of the puzzles, the picture will emerge. And then the last bit which I want to bring it to your notice is that kidneys, when the liver cleans out the A beta like protein, then the kidneys also have important function. We still don't understand how kidney function can be improved with neurotonics, or are there neurotonics which are targeted to kidney function and thereby helping the brain to function better. Similarly, the Deepana Pachana Dravyas, or something which is actually good for digestive system, how does it help indirectly the have Medhya kind of property, or which can help the brain and the nervous system function indirectly. So these are the aspects which we all need to, the people who are working in the transdisciplinary area can always think about, discuss, and design. Then we can design the experiments accordingly, or design the clinical or the preclinical or the basic research, biology research. We can plan it very well, and then all the findings from all the levels, like findings from clinical versus the basic biology level, I'm sure will converge and give us much better insights into the biological phenomena which are happening, because finally the biology is same, the body is same, and we are just looking at it in different ways. That's so with that note, I would like to stop, and if there are any questions, or even later whenever there are questions, I would be or queries or discussion points, I would be happy to answer those. And I would like to thank CAR and both Dr. Joshi and Dr. Yashaswini to give me this opportunity. It is very nice to connect with everybody on this platform, and I'm delighted to be here.
Yeah,
Thanks.
Thank you so much, ma'am, and it was wonderful presentation. Actually, the Ayurveda biology topics are very complex for understanding, because for us as Ayurveda clinician and academician, it is like a alien language for us. But the way you presented this article and the way you simplified it, it is now easier to understand in terms of the basics also. Fundamentals of Ayurveda, and in terms of the biological principles as well. So it's a very interesting way you are putting up the whole thing together. And I would like to mention this to all the participants that majority of the Ayurveda biology related researches are happening in the neurosciences, biology or biophysics laboratories. Even this article is also originated from those labs. Uh, but now it is high time that as a clinician, we need to come forward and put our remarks that what kind of a design is adequate to prove some of the efficacy. And disease modification, not only in terms of prognosis, but reversal is also possible with ekal drava chikitsa, like we call it. So single herb medications are
Yes,
totally relevant, and they can be useful if we have such mode of action data. And even
yes,
for students in future, they will be more related to this. It becomes more scientifically evaluated when they can see what is happening to the medicine when it enters the system. And not
sure.
only in terms of the disease modification, but you also explain a lot of things about how the toxic elements are getting excreted and how you are highlighting the importance of gut microbiota. So many inputs you are giving in this presentation. So I hope this will become inspiration for the future researchers and take it further. So I think there are no queries here, but I will just ask one basic query. Many of our clinicians, they wish to collaborate on biology themed, you can say, projects in the laboratory, but they are not aware about the models, which one to take, or even while reading the articles, we are not aware which one is the best suitable model to follow up, you know.
Yeah.
So would you please comment on that?
Yes, so that's actually a very dear subject of our institute and our department. So if anybody is interested, we can actually we have done a course earlier, and now if COVID is really gone, then we can also do the experiential, means they can come to the lab and actually see what model organisms we are using. We are actually in my group, we are even using a tinier organism than this, very tiny organism called C. elegans, but you can see how much information we can get from that. One, so we have many models. So like this Alzheimer's models per se,
Hmm.
I had a thing that it is present for, it's there in rat and in mice, not in the rats, mice. Then it is there in Drosophila, C. elegans, zebrafish. Then there are lots of these kind of models which are available where you will see different cell lines. So you can see different aspects we can study at them. That's what I
Hmm.
say, that if you keep, see, one dish doesn't make a meal, right? So you have to have all the dishes kept together, and that makes a meal.
Hmm.
So that's how we have to keep in mind that each model is giving you one important information,
Exactly.
and design your experiments like that. Either, see, two models will give two different information. You can put them together and make good piece of information, or something which will be, now if we are saying this, for example, I'll just give you one example. Now we are saying there are probiotics, right?
Yeah.
Then there are hundreds of such biotics, like good bacteria, say not
Yeah.
hundreds but at least tens of them, right? And they are making something which is good for brain. Now,
Yes.
if each one of those have to be studied, then my worms will come into picture, where we can feed one bacteria and that worm can live happily on that one type of bacteria. Then we can
Okay.
actually start dividing and say that, okay, this bacteria does that, that bacteria does that, this bacteria does that, and all put together, what will happen?
Yeah.
So this is how we can actually start combining the pieces together, and
Ex-
that kind of thing is available. You have to zoom in. I keep saying that zoom in and zoom out. So zoom out, complete good zoom out is Ayurveda. It has given us a very beautiful picture on the functional level. Now, if we want to understand the bits and pieces of it, the big picture is always there. The bits and pieces of it will bring our confidence up, and
Exactly.
most importantly, it will start telling us what we can do for wellness and not for the illness alone, and
Yes.
how we can stop Alzheimer's coming up. That will be possible if we take a closer look at the things by with the help of model organisms or model systems like cell lines, and
Yeah.
that's the advantage.
Yeah.
So you can actually contact us in our institute itself. we actually are not dealing with mice and rats, but ourselves, but we have collaborators. But
Yeah.
in our organization itself, we have C. elegans, we have Drosophila, we have cell lines. So these
Okay.
are the three systems which we use, and we train people. And yeast, we also have yeast, and we train people. We can train interested people on that, and we can have collaborations. And we also have a hospital, so we have both the clinic and the clinical research and the basic biology research and chemistry in the very same place.
Hmm. Yeah, and it's very interesting to know that. And you being here is also very necessary to tell us that, okay, we are ready to collaborate. Because sometimes the participatory research is also important. That where these organizations are working, various organizations are working, independent in the independent verticals, and they are not at
Yes,
all aware that what is happening in the other
there's no
organization.
crosstalk. Yes,
Yes. So this is a good platform of Council for Ayurveda Research, and we are definitely working on this model that with the Journal Club, this activity becomes just like a tool where we can introduce such systems to the new researchers coming up, and they will also understand what to read. Then only they
correct,
can think what
correct,
can be published and what can be done.
correct. Yes,
So
yes,
it's very
yes,
interesting area, and I would also like to mention that the new developments in the academic section are coming up from the ministry, and they are also considering Ayurveda biology as integral part of
yeah.
BMS course. So
Yeah.
that is.
So what I should tell you is that we have started a program
Mhm.
on MSc in life sciences with Ayurveda biology as specialization, and
Yeah.
we do have one good set of very transdisciplinary group of students, where group from coming from the pure sciences and Ayurveda graduates, PAMS, all
Okay.
are studying in the same place, and it's a very nice environment. It's very nice. Their whole 1 year, second year will be in the labs, and the
Okay.
first year will be mainly classes. So we have the first batch running, and that's going on very well. So we have the second batch coming up. So I would take help from CAR to
Yeah,
reach out to
definitely.
people and
Definitely.
say that we have MSc in Ayurveda biology, MSc life sciences, specialization is Ayurveda biology,
Okay.
and you can I will definitely require help from CAR to actually give us some visibility in the
Yes.
student population in the Ayurveda area. Yeah,
Yes, definitely. Thank you so much, ma'am, for being here. And it was wonderful presentation. You really decoded so many technical aspects into this, and this article is now all the more clear to us.
yes,
So thank you so much for being here. And
thank you,
I
my
would also
pleasure.
like to mention a special thank you to all the participants. Yashaswini is here, and Dr. Asmi is here. So thank you for being here. And I would also like to mention that this recording will be available with the Facebook page of Council for Ayurveda Research. And if there are any queries, you can always get back to us at Ayurveda Research USA at gmail.com. So with this presentation, I hope that many new comments might come up, and new inquiries might come up for this course also.
Yeah,
So thank you so much for that inquiry and that inputs from your side. So
yes.
uh, looking
Yeah,
forward.
yes. I just would like to ask you one thing.
Yes,
That you think at any point how we can make it like the presentation or the suggestions or even the journal club, how
yes,
we can be
definitely.
of help and how we can make it better, and so that to cater to many more people coming from many more different fields,
Yeah,
so to say. And even in the in Ayurveda field, there are so many different aspects. Bhaishya Kalpana is there,
yes,
Rasa Shastra is there. So many different aspects are there, and we can actually see how we can make it. Like you can tell us
yeah,
from we are coming as a biologist. You can tell us how better we can do, how what more we can do, so that
yes,
things will be more clear to the Ayurveda fraternity, and we definitely learn a lot from you. The whole concept of Medhya, I think, whole of my life I can learn from all the Ayurvedic experts on that. So
Exactly. And it's a win-win condition for both the faculties,
yes. So that's what I should want it to be. Want it to be. It has to be really a win-win situation. It should not be. It's a level playing field.
Yeah.
So there is nothing high or nothing low. So we all need to collaborate. And yes, Ashwini, you can also suggest if there is anything which we need to. Dr. Asmita, you can also let us know, and I will be happy to take comments and the questions even later point. Yeah.
Yeah,
Thank.
sure.
Thanks.
Sure. Thank you so much.
Yeah.
Thank you so much, ma'am, for being here
Yeah.
and also inviting us and our proposals to TDU.
Yes.
And we are sure now that you'll be there to support us. And
Yes.
together we can plan out many projects in future as well. With CAR, we are always there. And you being editor with one journal yourself, you can also suggest some of the topics for the journal club where we can introduce such newer branches where integration
Yeah.
is possible into basic sciences and Ayurveda. So that will be
Yes.
really interesting to explore.
Yeah. So my colleagues also, I would ask them to get in touch with CAR because all of us are doing that kind of research. So
Yeah.
we will be in touch with CAR.
Yes,
So
yeah,
that's,
definitely,
yeah.
definitely.
Yeah.
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 Thank you so much, everyone. And
Thank you. Yeah.
we close for the day. Thank you so much, ma'am, once again. Yeah.
Yeah. Thanks. Thanks. Yeah.
Thank you so much.
Bye.
Yeah.
Bye.
Thank you.
Thank you, Yashaswini. Thank you.
Thank you. Thank you for this wonderful presentation and also for your open invitation.
Thanks.
It
Thank
is
you.
your
My pleasure.
presentation was really, really wonderful, and it opened up so many aspects, and it was really
Yeah.
it was really
Thank you.
absolutely great. Thank you.
Thank you. Thank you.
It's great to
Thanks
have
a
you.
lot.
Thank you.
And let's catch up more often. Yeah.
Yes. All
Definitely.
right.
Thanks. Yes.
Yes.
Yeah.
Okay.
Bye bye.
Thank you.
Namaste.
Bye. Namaste.